Hello, everyone. Thanks for joining us today. I'm very happy to introduce JP, the Dr. Jen Pan Huang. JP is an associate research fellow at the Biodiversity Research Center of Academia Sinica, which is in Taipei, Taiwan. His research focuses on speciation and conservation genetics, and his work with a wide range of organisms like lichens, beetles, orchids, and spiders. Despite being an early career researcher, he has already published nearly 60 papers. JP was also a postdoc here in the Field Museum, where he worked with Dornstein on lichens diversification. That's how many of us got to know him. But today, JP is going to tell us about his work on beetles. So welcome, JP.
All right, you guys can hear me, right? Okay, thank you. It's very nice to be back here, and haven't give a public talk for a long time, so hopefully I still can do it. And if you guys don't understand me, you can just raise your hand, I can answer, right? So like Alejandrina said, I was a postdoc here working on lichens. I still work on lichens. I host Thorsten and Matt back in Taiwan 2 times, right, already. And if you guys want to collect beetles or lichens or like butterflies in Taiwan, you can contact me and we will be welcome and then help you with setup with the field trip. All right. But today's talk is actually about beetles. All right. I hope you guys will like it. So my work mostly is about biodiversity because I stay in the Biodiversity Research Center in Taiwan. So we care about biodiversity laws. And this is kind of a global, it's not kind of, it's a global trend, right? Like this very influential paper that somebody went back to the same place to collect the same thing, but they realized the biomass lost a lot. And this is not just a global thing, it also happened locally. So these things are written in Chinese. So basically, there's a lot of loss of species diversity and biomass also in Taiwan. And then in the other example, so this first species here is also they almost went extinct in the past. But recently there is a rebound in population size. But when you do the genetic to calculate the effective population size, they're kind of still small. So you don't know like how long will you take, even if you have a census population size to be back, you still need time for the genetic diversity to recover. Okay, so that's why it's not out of the vortex yet. All right, so with what I can do using genetic data, because I used to study speciation genomics and I also like systematics, maybe I can use my expertise in genetic data to help doing some conservation work in Taiwan. For example, we use genetic data to determine how many different species or how many different units are there, because sometimes species are difficult to tell using our eyes. Or maybe you need to find the right structure, but you haven't found it yet. The second part is that using genetic data from an evolutionary perspective of view, we can use it to reconstruct the history. So we know that why do we have so many different things here? It could be ecology. It could be also driven by geography. But at the same time, using the data, you can actually use them to help us understand the conservation priority of different things. Because now genetic data can help us estimate the change in genetic diversity. And then also some more recently, people are promoting using genetic data to help us estimate the effective population size. And one of the most important one, or mostly frequently cited one, is the 50-500 rule. So if that's like, if you have an effective population size smaller than 50, it means that your species or your population are going to lose genetic diversity very fast, rapidly. which also means they will not have the evolvability facing rapid future climate change. And if it's below 500, there is no immediate short-term risk, but long-term, you will still continuously lose the genetic diversity. So these two are very important parameter value here. Okay, but when I say genetic data, there are also a lot of different type of genetic data, right? Everybody here knows that we can use Sanger sequencing. You can also, a lot of people use RAS-seq. And now, if you read a genetic or conservation genomic data, people start to promote it that we need reference genome. But do we have enough empirical data to support? If we have a reference genome, what additional data does it tell us? We don't really have that yet, because either you have reference genome and then people move on to use reference genome to do a lot of analysis, or like invertebrate, if we don't have reference genome, we stay with a lot of population genetic study. So that's one of it. And then, so basically, my talk today would be like using genetic data to help us understand the evolution and history of a group of Sky Island beetle, and then also at the same time use the same data to help us understand the future conservation plan for those beetles. All right, so this is my study system. It's a species complex, so there are currently, there are 3 populations on 3 mountaintop of Taiwan, like here, here, and here. And this island is Taiwan, in case you don't know where Taiwan or what Taiwan looks like. And it's a very mountainous island. It can be up to like 4,000 meters high in the middle. But these 3 beetles usually live in a range around like 2,000 meters above sea level. So in between of the populations, it's like valleys or gorges, so it's not connected physically. And Asian beetles, especially like me, we like to collecting beetles as pets or as personal collection because they look nice. So they have been under a lot of collection pressure because it's also the first stag beetle species every year to emerge first. So people will like to just go out and collect them to see the first sign of summer. And some of the beetles, their habitat has also been underwent a lot of habitat destruction. And it's different across the 3 different beetle populations. So at the same time, I also want to know that if there are different levels of human exploitation, do we see that in genetic data? And these 3 beetles are actually a good system to test it. Okay, so why I choose Sky Island? So the first thing is that in the future, maybe because we know that at least a lot of people are hypothesized that because the climate gets warmer, things start to get like either you go to higher latitude or altitude, like you move to like your suitable places. So it's a place that may be affected more by future climate change. And the second thing is like it's important for me, it's easier to define population because it's not connected. So if you collect on one mountaintop, it's one population, the others is different. The third part is that there are not that many model parameters, so it's easier. So if you only have 2 populations, you have 2 population size, 1 divergence time. Maybe you can add 1 or 2 migration band. That's it. Easier. If you have too many species, then just difficult. A lot of time you don't have enough data power to help you estimate a parameter. And the third part is, like I said, on those different mountaintop, if you have a good governmental record, you can, there may be some good historical records for different level of human interference among the different mountaintop. For example, in this figure, the middle one, the forest was chopped down and then being turned into cabins. And then if we have a record saying that it happens in the 1950s, so you know that it happens in 1950s, and it's different from the other 2 populations. So you have a comparative system to study whether human direct exploitation has any impacts on their genetic structure. Right? And it's good for my system here because Thorsten and Matt went to this place before, and this place was a very big mountainous forest area, But after the Chinese Civil War in 1949, there was a settlement of veterans into the region. So they developed them into farmland. So a lot of them just got like clearing like this, okay? And we know exactly the time, like around the 60s, everything started to happen. And then we also know that in Taiwan, we have a forest protecting law started around the 1990s. So there are different settings of human direct interference with the habitat. And for the other two populations, so this one is a newly discovered population. It's hidden in a national park. So it requires like 3 hours of walk and camping to get to that habitat. So it's probably being isolated and protected for a long time. And then the one in the B column, the one from Hsinchu, it was also in a native people area, but recently it's being developed into like glamping and camping regions. Okay. Right, so we know that something happened in their habitat, and there is a good historical record. So the first thing that I did is use this system to study like how many things are here and why do we see different things here, and then do these three different things require different conservation planning, or you can just protect them as one, all right? Okay, so in the following, there will be 3 case studies using exactly the same system. But in the first one, so it'll be a chronicle, in a chronicle way. The first one is when I just got back, I got back to Taiwan in 2019, and this paper published in 2021. So it's our first paper about a system. At the time, we only know 2 populations. So the one that's very difficult to get to in a national park, we didn't know that yet. So we only have these 2, and then we use Sanger sequencing, 1 mitochondria and 4 or 5 nuclear loci to study their evolutionary history and genetic difference. And the first one part is using barcoding. Often, if there are different things, it'd be easy to identify them as different species, right? If you only use 2 or 3 individuals, barcode give you good result. But we collect 40. So when you collect 40, you will always see this. So this one's supposed to be from this population, right? It's a cluster with the other population. So what would you do though? If you don't remove that individual, you don't have barcoding gap. If you remove that individual, like if you're like a scientific ethic is not that good, then you can just show that yes, we have two good population because the barcoding gap is actually pretty big, yeah. But anyway, you're saying that these 2 are different things, but somehow they may still have some historical incomplete lineage sorting there in the mitochondrial data. And it's not just in the barcoding. If you use the nuclear loci, and then you can use BPP to help you estimate the genealogical divergence index. And this index has been developed for a long time. Usually, if the index is smaller than 0.2, it means the intraspecific differences. If the index is larger than 0.7, so the index range from 0 to 1. If it's larger than 0.7, it usually indicates the divergence between the 2 things that you are testing are different species, or at least when you ask a taxonomist, they will tell you these 2 are different species, usually, okay? And our data is cool because it's located in 0.5. So it's something that you don't know how to call them. So they are different things, but they are not that different that people are sure they are different species, but they are certainly not the same species. So just call them whatever you want to call. So I just call them the 2 different things. So we have 2 different things, okay? And then the third part is that with the mitochondria and nuclear loci, I cannot do much, but I can still do model comparison using DIY ABC. So I come up with 5 different models. So these different models basically, it could be like a northern population origin and then colonize the central part, or this is the original population that colonize this one. Okay, so that's the first 2. The other 3 is that just there is an ancestral population split into 2. Or maybe there The first two also involve like intermediate gene flow between the, okay. So the best model from the Sanger sequencing data is saying that there is a ancestral widespread population split into two, and then nothing happened after that. Okay, quite boring. So we have two things, and it's Sky Island, and then split into two different things, and that's it. So then what about morphology? In stag beetles, we like to see their mandibles, right? Because they fight in the cool. So when we use morphometric to study the mandibles between different male, they do seem to have, they do seem to occupy different morpho space. Okay, so the red one, the blue ones, they are some like in between. But if you do a statistic test, that's significant. And there is also a different thing. Like if you look at the northern one, it's a little bit brownish red. The central part one is a little bit darkish brown. But when we do the RGB, so you can extract the RGB value from the picture, there are kind of a lot of overlap. So if you look at the body coloration, there is some like a degree of difference, but it's not significant. But when you look at the mandible, it's significant, but it's not like very, very different two groups. They are still very close. Okay, so all the evidence here suggests these two are different things, but they probably diverge very recently. Okay, so going back to the first several slides, what can Sanger sequence data tell us at this part is mostly about evolutionary history. They can tell us that these two are different things, though they probably require their own conservation plan. And also, they're probably isolated by physical barriers. And then there's no gene flow after that. But there's always the real guy that cluster with the other part. So if you sample more, there's always the real guy that will pop up. Okay, all right, so that's the study one. And then because the first study wasn't very informative to me, so I moved on to use DD-RESIC data to study the same system again, but at the time, 2020, we discovered a middle population. So this time we have three population or three taxa here, and we use DD-RESIC, and then at the end we come up, we retain about 2,000 loci in this study. And this time, using 2,000 loci, basically the three things are three things, okay? I don't know why that, what can you call it? They're just three very different lineages or clades. And then on the PCA plots, they are just far away clusters. And then in the mixture part, there's no mixing. And then when you do any AIC or BIC estimation for the K value, it's always three. Okay, so three things, nice. But we have one weird thing, right? In the mitochondria data, we have one that from this species, but it's clustered with a different population. But in this data set now, it's actually very clear this individual is 100% from its population. So probably the mitochondria data, the result is just because of ancestral lineage sorting that's incomplete yet. All right, and why do we know that? Because if that individual is a hybrid, this program that we use will supposedly it will show some signs of hybridization, like so each line here is individual. So if somebody is hybrid, it will show like a red color with not only its own population but also the other population that give some of the allele to this individual. And we don't see that for this individual. Okay, so probably, like I say, three things. There's no gene flow after their divergence, just three Sky Island things. Pretty neat. And then the mitochondria data is probably because incomplete lineage sorting because they diverged quite recently. All right, and then we also use some other program to test for migration, and then the best model is still without adding any migration band. So you don't need migration to explain the data that we see today. But what's good with the RESIC data is that if you have a good background about the overall genomic rate of mutation, you can estimate their coalescence time. So this time we can estimate that these two are sisters, and they probably diverged about 200K to 200,000 years ago. And then the older divergence is this one to these guys. That's about like 600,000 years ago. And what's more interesting is that if you also use the same data to reconstruct the ancient demographic history, you will see that around like 600K years ago to about 400K years ago, the population size was big. And then there is a sharp decrease. So that probably represent the first split between them. And then around 200K to until very recently, there is another plateau and then sharp decrease. So they're probably just one very widespread population. Every time there is a environmental event, they just shrink to mountaintop and then stay there and then didn't move. Okay. But with RESIC data, because if there are more and more studies showing that you can use RESIC data or genomic data to help you do model testing, if there is a very severe human impact, sometimes you will be able to detect them using RESIC data because the statistical power is huge. So we use FastSim code to simulate the 5 different history here. So the first one is the continuous decrease in population size because we know global decrease in insect population, right? So it's just a pattern that everybody's talking about. The second one is specific to our system because we know this guy, their environment has been heavily exploited in the 60s. So we expect there may be a change or decrease in effective population size in around that time, and then around the 2000s, because camping and glamping become a big thing, so all the environment got developed. All right, but and then there are like the other models. The last one is kind of the sanity check, so nothing changed. They stay the same population size. But to our surprise is that for all 3 population or all 3 lineage here, the best model is the first one, continuous decrease. So they just keep decreasing, all right? No matter how we filter or sample or consider different missing data, the results still the same. The 3 population experience exactly the same history, okay? All right, so the take home from study 2 that used RESIC is that there are 2 different things, but we can happily cross that there is no weird thing. That weird thing is probably just the ancestral polymorphism. And then we know the origin is because of habitat fragmentation that becomes sky island species. But we don't have any evidence supporting recent human activity leave any tractable marks in their genetics. And this result bugs me a lot. I really don't like it because it's obvious. Like if you see clear cut of a mountain, how could they leave no trace in the result? So in order to further test if it's a real thing or just because the data that we use, DD-RESIC, still didn't have enough power to detect this impact, we move on to use reference genome. So what I mean by reference genome is that we use nanopore to do the long read sequencing and then assemble them into 8 nice chromosomes, but we don't know which one is the sex chromosome. So we using red tag to map them back to the European stag beetle. So we have a good chromosome or like a pseudo-molecule level, 8 chromosome level genome. And then we use the original RESIC data that I used in the previous study, map them back to the reference genome, and you see it's nicely. So RESIC is actually good. It's a good representation, like randomly across the genome. So if you use RESIC, this is a good one to show you that you are not just sampling some part of the genome, okay? But with this data, it's actually it will give us some more nuance of the data that we have. Because, for example, when we do the red-seq analysis, we kind of assume they are unlinked because we don't know where they are. But now with the reference genome, we can map these red-seq loci back to the genome. So now we know where they are. And then people also study Beetles recombination rate before. So with the recombination rate, you can also estimate how often do you see the heterozygous across the genome is. And then it's important because people use the same method to study the run of homozygosity in, for example, mammoths before. So mammoths used to have a big population, but when the climate change, when the glacial period passed, they kind of shrink to a small island, and that small island population has a very long run of homozygosity across the genome, which means they are all inbreeds. All right. So this would be something that we won't be able to see using red-seq data. But with this reference, maybe we can see that. So we actually saw it. So it's nice. So the middle population here is the one that we found in the national park. It's been hidden for a long time. It has very low level of runoff homozygosity, which means the population has been very stable. The other two populations that people know for a long time, especially the one that's been heavily exploited, it has longer run of homozygosity. Right. And then this is the previous result. So we used site frequency spectrum to estimate the demographic change in the past. We can only estimate a long time one. But recently, there are new models being developed. For example, there's a model called GAN. You can use the linkage disequilibrium with the heterozygosity information in your genomic data set. So your genome using Plink, and then you can use it to estimate very recent demographic change. For example, this is a wolf example. They have a census population size change, and they also have the estimate effective population size change. There is a time lag, but still match pretty well. And this program GAN, they just published GAN 2 this year. So in the result that I'm going to present, we still use the previous version, but the GAN 2 come out now. So it's supposedly will be easier to use. All right, so this is our result here. The blue line is the one that the population that's being exploited after the Civil War. And you can see the time of decrease. You probably cannot see it. So it's around like 80 years ago, so 1940 to 1960s. There's a drop in effective population size. So now we see it. It's cool, yeah, and I kind of feel good, but also bad. I don't know if that's true, but at least with the reference genome, it gives us more nuance, like the recent change in demography is different from the ancestral, like the ancient time in the past. Okay, but why is it, right? So my explanation in the paper is that if you look at the site frequency spectrum one, the one that we published in the second study, we know that the effective population size changed from like 500K to only 100. So that's like more than 5,000 times difference change in the effective population size. That's a very significant signal in the genome. Whereas in the recent change and the rebound in population size, that's less than 10 times difference. So probably if you don't have a genomic data, if you don't have a genome reference, you only have the mutation spectrum, then the only signal you will pick up would be the most significant one in the past. You probably won't be able to pick up the recent one. But with the linkage disequilibrium information adding into the data, maybe we can start to pick up these nuances. Okay. And also with the linkage map, there are also a lot of new programs being developed right now or method. Any estimator is old, but now it has a specific function that can only help you estimate the linkage disequilibrium between SNP across different genomes, so SNP from different genomes, to help you estimate the effective population size, because usually linkage disequilibrium happen in the physical linkage. But if you have a very small effective population size, it kind of everything it behaves the same way. Okay, and so in this result, the one that's been heavily exploited before actually has the largest current effective population size. But it also fit our expectation because that population is very easy to collect. So people just go there to camping and then collect the beetles. And the one in that's historically stable, but in the national forest, it actually has the smallest effective population size. So it gives us a very difficult part. Is historical population size stable, but it's not big. But there is one that's being big, but genomically it's not healthy. Okay. And then finally, with the reference genome, we actually, because I didn't expect this, but you can always do this, right? Just plot the FST and genetic diversity pi, and also the FIS is the inbreeding. So we actually find a genomic region here that shows like through the roof high level of inbreeding within population, and also a high level of FST. So it's the difference between populations. So which means these three populations on like chromosome 5, the first part of the chromosome 5, there may have been a selective sweep. So one of the population, especially the one in the very north, it may have adapted into a different environment or using this part of the genome to do something. We haven't done any annotation in this genome, so we don't know function. But based on the result here, we know it's probably there is a selective sweep happening somewhere in that genome. So these three things are not just three different things. They are three different things with local adaptation. All right. So the take home for the final part of the third talk is that we know there are three different things. They need three different management plans. But with the genomic data, we know that there's probably some selection or local adaptations happening inside them. So which means that you should not put them all together and mix them as one. And then the part that made me really happy is that now we have evidence to show that human exploitation of the habitat actually leave a mark in the genetic data, but you sometimes have to have a lot more data power to detect it. And then based on the 50-50 rule, like they are probably okay or not okay, but short term, maybe one population is not okay, but the other two populations kind of okay, but long term, they may not be okay. It's like at a very weird stage. They are not protected, but you should not mix them. And I don't know what to do with them. So you probably just tell your government, just don't do anything about them. So yes, so that's the result. But anyway, the important thing is that when we write a grant or a proposal, that sometimes you want to argue that you want a reference genome, right? So if you write a proposal saying that you really need a good empirical data to help you write a strong argument why you need a reference genome, please cite my paper. Okay, it's right here. You can directly saying that a comparison give you a different result. But it's also a time that I realized I should move on, not stay with this system, because the life history is long. It needs 4 years of development to become adults. And yeah, it's just difficult. Yeah. And you cannot do anything. But so if you cannot do anything, the government just will not spend any money. So that's actually the reason I'm here. So I also study the long arm beetle, and it's big and protected. And we also know that it become a protected species since 1989 in Taiwan. Okay, so we have like a old collection before 1989, and also the new collection. And from those, the fresh collection that I did a couple of years ago, we found that the K equals to 1, so which is great, because if we can find some population has higher genetic diversity, we may use them as the reservoir to release them to the other plot that has the lower genetic diversity. Like if you have 3 different things, you cannot do this. But if you only have 1, that's great. You can do something. A government will give you money to do something. Okay. And what's even better is that I also get a funding for this. So the next 3 years, I will have funding to do conservation genomics of this species. And so in the study, we will also do something similar in Vietnam, because Vietnam also recently underwent very rapid development. So there are national park. There's also a lot of habitat being destroyed rapidly. So we will do similar things, compare the genetic diversity within and outside of the national park. But in addition to the contemporary data, we will also have this species is good because people know it for a long time since Wallace. So we will also have historical data. So it's not just spatial variation. We will also have the temporal diversity in genetic variation. And what's even better is that here in Field Museum, I found one that's collected in 1961 from Taiwan. And in addition to that, the other closely related species has been identified by Ronald Young. So if I'm not good at identify species, he already done that for me and also run a review, so I won't have wrong identification. So that's also the reason I'm here this year. I will borrow this specimen back, try to extract DNA out because we have a reference genome now. So we just use the shotgun sequencing and then map them back to the reference genome to fish out all the SNP to see if we see geographic variation in genetic diversity or temporal variation in genetic diversity. And also in addition to that, now I also do a lot of butterflies. I did not do butterfly before, but because Taiwanese government is really fixed with butterfly because they are beautiful. I know that Matt and Thorsten, we went to the southern part of Taiwan and we've been waiting for one of the butterfly to fly out, the yellow one. So that one is also a protective species. So now in the whiteboard in my lab, it's a lot of butterfly sequencing genome, like nanobore done, high-C done, and then resequencing done, and then, yeah. All right. So that's it. And this is my lab. So the study, so my first research assistant, he's now a PhD student in Northern Arizona U. And the second paper is done by Mattia. He's now a postdoc in Germany. And then Jeff, my most recent graduate, PhD student, he's now a postdoc in Tokyo, University of Tokyo. And then the other of my team and also my wife and my kids, they helped me collect some of the data. Right, that's it. Thank you.
We have time for questions.
Yes, Bruno.
Cool.
I mean, even if they're technically okay, like, ineffective for 50 or 100 for an insect, for me, it's astonishingly low. Like the things I work on, whenever I estimate this, I mean, hundreds of thousands or more. Is this typical? Like, I can imagine, so the areas, all those Kai Islands must be super small, and like, are the census populations also super small? Or is this mostly like due to the genetic bottleneck and the census population is kind of okay? Yeah.
I think it could be both, but usually, because we have a very good beetle amateur population in Taiwan, and then if they went out to collect the beetles every day, like if you have a good day, you probably can collect 14 of them. And annually, they only come out like 1 month per year, particularly within 2 weeks. So if I will say the census population should be below 1,000, like a couple hundred to 500, if you collect them continuously throughout the season. So it's not big. The population size is not big for the suite. That's why I choose them, because it's small, so it should be easier. And at least I originally thought so. So it's become too easy and different. Yeah. But yeah, thank you. Yeah, this is a species with small population size.
Someone more?
Yes.
When you're like using the 5500 method or rule, what are the specific like parameters you're supposed to set like location size and like length of time?
You mean the effective population size?
Yeah.
Um, so there are many different ways to calculate it, and here in this study, the only thing that I ask the program to do is to choose the variable site from different chromosome, so that we can see that how often do we see the variation between the two different genes. The frequency, how often do they covary together? Yeah, if they are from different chromosome, because if they are from the same chromosome, they probably will vary in the same way. Yeah, but if they are from different chromosome, but still very often change in the same way, it probably means the entire genome is under the very strong recent bottleneck. Do I answer your question, or?
I was kind of more asking like the collection methods.
Oh, the collection method.
Is this that rule entirely just for analyzing the genetics, or
Yes.
does it okay?
So
Yeah.
that's mostly for the genetic data, but it has some assumptions like the sample that I collected are equally related to each other, and then it's a randomly mating population, so that you can have a good representation for the whole population.
Yeah, I was asking more about sampling.
Yes, yeah. So I collect 30 of them, and I assume they are randomly sampled from the population. Yes. Good. Thank you.
More questions? Someone?
I thought that the part where you talked about the chromosomes and the FIS and the FST values was really cool. I was just curious if you had any like guesses of if you did take a look at the genome annotations, like what about that chromosome is why it's like adapting more than the rest of them?
I don't know about the chromosome, but I know that the population in the very north, they come out at a different, they become active at different time of the day. So for example, the two in the middle part, they come out early because it's hotter, so the temperature rise faster. So you usually collect them around 10 o'clock in the morning or 11. But the one in the north, because in the morning is usually foggy, so people collect them around 2 o'clock in the afternoon. So the time of become active is a little bit different. Yeah, that's something that I can say that I know about their ecology from what I know from the beetle amateurs. Yeah, but I don't know about the genome yet. I also don't want to spend too much time now in this system because it takes too long for them to develop. But maybe one day if I have too much extra money, I will keep dig deeper. But yeah, thank you.
Thank you. Someone?
I have one more. So with the super high resolution data, what did they discover about the population in the national park? So is it constant? Is it going through fluctuations?
It's going through fluctuation, and also something that we didn't say in the paper is that population has been exploited during the Imperial Japan period, and we also detect a decrease. So it's the yellow line. So it's prior to the Taiwan, the island of Formosa becoming back to it's still a sovereign island, yes. But so after the Civil War, yes, but during the Imperial Japanese era, there was also a decrease in effective population size. Okay, and then it become hidden. Nobody go there. Only the native people knows where they are. So you can see there is a decrease in effective population size. The most different one is actually the northern one, the one that lives slightly in a different habitat. It's always stable, the green one, but also recently, because the habitats started to become developed into glamping and camping area. So everybody drops.
More questions? No more? Okay. Thank you so much,
Yeah,
JP.
thanks a lot.
Thanks everyone for coming.